Well, good afternoon, everybody. Welcome, and I'm speechless, as you can see. So this is Residents Present, and today we have the great privilege of having Blanche Alter talk to us, and her title is one that will intrigue everybody. What are all the words, and what did I do that used those words? And Blanche is an eminent physician and researcher, and she went to college. Well, first of all, she grew up in Toronto, across the border, so at least she wasn't liable to the draft. Anyway, then she came and went to college at Radcliffe College, and then went to medical school at Harvard Medical School, and did her fellowship training at Harvard Boston Children's Hospital. And her mentor, one of her mentors, was David Nathans, who many of us consider to be probably one of the leading lights in 20th and 21st century medicine. Anyway, she worked at several institutions. She got an MPH at Johns Hopkins and worked at Hopkins. She worked at MIT and worked at UT Galveston. And I probably left out 2 or 3 places, but she did most of her work at NCI, or National Cancer Institute. And she's going to tell us all about it now. Thank you.
Thank you. I promise, if I remember, to repeat questions, but that's a few minutes down the line. So I was challenged to come up with a useful title, which I hope was why some of you came. And I did that partly because my career most recent, before coming here, was in public health. And so there are a lot of vocabulary that came out, sorry, that's the Canadian in me, came out in recent years. So that's part of what I did here. I'm only going to tell you a little bit of what I really did because that's boring. So the first thing is that our trainees at the NCI gave us all an award. Happens to be a paper plate, but that's the award. And if you read it really carefully, it's most likely to catch, C-A-C-T-H, typos. And the inside says, here that you're a professional booger, no, blogger. So I was well known amongst my trainees and colleagues for editing. So if anybody says anything they want edited, I'm happy to do that even if I know nothing about what it says. Now, I used to, at this time of year, give a lecture at Camp Sunshine, which is on Lake Sebago, which is 2.5 hours away from here. And it was for disorders that I study. And I prefaced all my talks by pictures of birds that I'd seen recently. So I thought I'd do that for you folks. So anybody, just think about whether you know what this bird is. That's only for those who've lived in the South. It's a brown pelican, which I took this picture in February when I fled winter here. And the fact that Michael may have mentioned I grew up in Canada doesn't mean I appreciate winter. This picture was on the C&O Canal, which is near where I lived before I came here. It's called a painted bunting. And C&O stands for Chesapeake and Ohio Canal. But the painted bunting really is that pretty. It's amazing. And even during COVID, there were a lot of people gathered along the side of the river looking at this bird. Now, when I left a year and a half ago now, I guess a year ago, there was a party and they made cookies of reminiscent of the things that were part of my background. Whoops. So looks like my pointer doesn't work. Okay. In any event, there's a syringe and a white coat. When's the last time you saw a doctor wear one of those white coats? And a cookie that was a red blood cell, because that's what I used to study. So who am I? I grew up in Toronto, Canada. I went to college in the States in a small place across the river from here. Med school in Balmer, and for those who are from there know that the T is silent. Then recently as studying could be, I did my Master of Public Health at Hopkins. And I've worked at many places that are listed here. Hopkins was my training, MIT was part of my pediatric hematology, and then I worked at Mount Sinai, at UTMB Galveston, and at the NCI. So this is a picture of Camp Sunshine on Lake Sebago. It's near Casco, and unfortunately it's not open this year because it's where families go whose children are vulnerable. But this is a picture of the door that was built into the family center, and as you see, I'm the right height for the middle. You know, one of the fathers is on that side and one of the kids is on the other side. Now, who knows who this is? Everybody. That's Tony Fauci. And I have to say that he is still working, although he does need Secret Service protection. So there's a lot of words that we learned during COVID that some of us may not have used a lot previously. Science, epidemiology, eponym, cohort control, I will talk about those, pandemic versus epidemic, what are statistics, and some of these other terms on the other side. And there's a book about the epidemic, the flu epidemic of 1918, I think it was, and they really thought it was an amoeba when they figured out it wasn't a bacteria. But they didn't know anything about viruses at that time, which is where we are now. So here are the words that we had to learn that probably weren't part of our everyday vocabulary. Science. It's the pursuit and application of knowledge and understanding of the natural and social world following a systematic methodology based on evidence. So there's science over here, and then there's fiction over here. And we all know that this side, the science side, is where we want to believe, I hope. I like this cartoon. Thank you all for coming to this small enclosed space to discuss a deadly and highly communicable disease. I love these cartoons. And science class. Welcome back to school. The first thing you'll learn is how a virus spreads. Well, I think we all have learned these things. Modern life. I'll probably stay in school until I get my PhD. Sometime I'd like to feel smarter than my phone. And I've given up on that. I'm happy if I can find the thing, never mind be smarter than it. So what is epidemiology? It's the study of the distribution and determinants of health-related states or events in specific populations and application of this study to the control of health problems. And this is where we're at this point, which is if somebody tells you about frequency or who's got it or how long it lasts, it's based on epidemiology. It's based on analyses of cases and following them and gathering proper data. It's not made up, okay? A cohort study is a type of longitudinal study, which is an approach that follows research participants over a period of time, often many years. And I'll tell you a little bit of what I did along those lines, but had nothing to do with COVID. The control group are participants in a study who didn't get the experimental treatment, but are similar in other aspects. In other words, we can't compare outcomes, I'm trying not to speak too Canadian here, outcomes, to a group of Asians versus people in another country versus people at Newbridge, etc. It's It depends on whether the groups have any similarities. An epidemic is a widespread occurrence of an infectious disease in a community at a particular time. We all know the word flu epidemic. Well, now we're in the realm of COVID epidemic. But let us hope there's not currently a COVID epidemic at this institution, which is the only one that really matters to us, except for those of us who go grocery shopping. Endemic is something found in particular people or a certain area. The agent is always present. So there's a statement, complacency is endemic in industry today. And we're waiting for COVID to change from an epidemic to endemic. And periodically you'll see that on the news or you'll read it in the New York Times, and there will be groups that say, oh yeah, now it's endemic, we're not going to worry about it. Well, I'm not sure we're there yet. A pandemic is an epidemic that is worldwide over a wide area, crosses international boundaries, and usually affects a large number of people. So COVID-19 was declared a pandemic in March of 2020 by the World Health Organization because it was so widespread. Now, what are statistics? They're the science concerned with developing and studying methods for collecting, analyzing, interpreting, and presenting empirical data. Empirical means it's based on numbers. It's not created out of whole cloth. Two fundamental ideas are uncertainty and variation. Now, we can be uncertain about something. That's not the same as statistical uncertainty. I'm uncertain about a lot, but that's not the same thing. Any measurement or data collection effort is subject to a number of sources of variation. So, for example, if you want to say the risk of COVID is thus and so in females compared with males, you have to understand that there may be differences in the group. It's a method for analyzing and drawing conclusions to tell a quantitative story. Now, an eponym is a person, place, or thing after whom something is named. So many of you may have heard the term Down syndrome. It has the apostrophe S because Dr. Down described it. It's trisomy 21. But it's not, he doesn't own it. It's not apostrophe, it's not his syndrome. So most recently, I would say in the last 10 years or so, all the apostrophe S's after the name of diseases have been removed. I don't know if that's just in my field or in your field. Okay. So Down syndrome is really trisomy 21, which means there are 3 copies of chromosome 21. It was described by Dr. Down, but it's not, he didn't have it. Some of the vocabulary is listed here. You've seen many of these words in the newspapers or on the evening news, if you don't eat an early dinner. But they all, if you don't know what they mean, you need to Google them. And I have made rounds with individuals more junior than I, and I've learned a very important term, Dr. Google, okay? And even in Mexico City, even with people who spoke English, if they didn't know what something meant, we consulted Dr. Google. A bacteria are small single-celled organisms that you can see under the microscope. And I don't have a, well, there is a picture of a microscope. I have to give you an aside, by the way. When I went to medical school, I had some scholarships, and if you were a scholarship student, you were not allowed to spend your money to buy a microscope, which is very unfortunate given that I went into a field that uses a microscope a lot. A virus is a submicroscopic agent that replicates only inside the living cells of another organism. So some of you early in the COVID epidemic may have seen all these YouTubes about how to wash your groceries and wipe them down and all that. They will test positive because if you're testing for the protein, even if the virus can't replicate, it'll test. So you have to know what your test is and what you're actually measuring. An amoeba is a microbe made up of a single cell that can change its shape. As you see, it's looks like a pair of gloves there. Protein synthesis, which is what everybody's hearing about, the material that makes up your genetic composition is the DNA. It has to be transcribed into something called RNA. DNA is deoxyribonucleic acid, RNA is ribonucleic acid, and then it has to make protein. And that's really the important part. The virus, the vaccines that are available now are mostly directed against RNA, which may mean they might work more rapidly than protein, but they're not against DNA. So DNA is this long word that that's why we spell it out. It's a long molecule. It contains the genetic code. It's a recipe book. It contains the instructions for making the proteins. It has a double helix shape, and there are 4 components which are listed here: adenine, thymine, cytosine, and guanine. Whoops. And as I said, the DNA has to be transcribed into RNA, which is then translated into protein. An RNA is ribonucleic acid. It's a molecule which is essential in all the biologic roles of coding, decoding, regulation, and expression of genes. And it's a complex compound that is important in cellular protein synthesis. And in some viruses, instead of DNA, they have RNA. So the coronavirus is an RNA virus. Protein is made up of different building blocks, which are called amino acids. Some of you, if you know people who are involved in muscle building and bodybuilding, they eat amino acids. Some of us are not willing to prescribe them for that purpose. But HB is an abbreviation that we use for hemoglobin, which is what I've studied a lot. And A is the usual adult hemoglobin. If you hear about something called hemoglobin F, that's fetal hemoglobin. It's the hemoglobin that the fetus has until it transfers over to the adult type. And then there's hemoglobin S, the sickle hemoglobin, which has a different amino acid at codon 6 with a switch from valine to glutamic acid. Now, genealogy, which I'm sure Carol will be expounding on at some later time, I'm not sure why it's called a family tree, because it starts with the parents, then it goes down to children, and then the grandchildren. Well, that's an upside-down tree. But in any event, that's the way it's described. And the chart shown here is colored in or shaded with the people who carry the relevant gene. And just for your future reference, we're about 80% women in this room. So women are the circle and men are the squares. I didn't develop that methodology. Carol, can you explain that? Well, yes, but
I don't think there's probably a
well, we like to make jokes about it. So a bacteriophage, which is a virus that lives within bacteria, as opposed to other viruses that occupy other types of cells. And I will say that I did go to college across the river, and one of the courses that I sat in on initially was Jim Watson's course on virology, except I was there the first day, which is when he won the Nobel Prize, which was exciting. But he announced that the only virus of importance was a bacteriophage, at which point I dropped the course. Okay, now this slide is a bit complicated. A chromosome is a string that has DNA and proteins, and at the ends of that, there are caps, which are a series of nucleotides called TTAGG, and they protect the genome during cell division. And the reason this is important, and this slide was not designed for this audience, is there are patients that I have studied who have a disorder in which their telomeres are abnormal. And that's one of the types of disorders that I will mention. But when the telomeres are too short, it means the cells have accumulated damage as they divided and may have accumulated some mutations that may lead to development of tumors. And I like this slide. Unfortunately, if you go online, the little yellow dots flash on and off, but I couldn't capture that on my PC. There's a lab in Vancouver whose name is called Repeat Diagnostic because they're analyzing the number of these repeats at the ends of the chromosome. And the lack of maintenance of these telomeres leads to genetic instability, cell crisis, cell death, and some of the disorders I study. Now, these are some words, and I wrote them down because I realized that they're confusing. One goose is a goose. Many are geese. But one moose is not a meese. And I realized I was giving a lecture, I'll never forget, I was at Ann Arbor, Michigan. I was giving a talk about the work with mice in my laboratory. And the plural, the singular of mice is moose, mouse. But to others than Canadian, it sounds like an animal with these big horns. So that's when I started talking about them in plural. Now, what did I do before I retired? That's what you really wanted to know. Besides sitting at home alone for a year on indefinite telework. But nevertheless, I trained in pediatrics, which I did at Hopkins. And then I trained in hematology, which is blood, not oncology, which is cancer. And I ended up at the National Cancer Institute anyway. So why did I do pediatrics and why hematology? I wanted the patients to be smaller than me. Well, the guy on my left, the shorter of those two, came up to me at one of the family support meetings for this disorder. And he was about 5'8". And he said, there's somebody here taller than me who has this syndrome. The syndrome is associated with short stature. I don't have that syndrome, but I do have the short stature. So that was one reason. The second is I wanted patients to have one disorder at a time. Well, that's wrong because, as Michael will reinforce, all these pediatric patients, they may have an endocrine disorder or a blood disorder or a bone marrow disorder, but sometimes they have all of them. And then, of course, I wanted to do straight hematology, which on some of my slides I used to call benign hematology, and my friends have made me change it to non-malignant. I'm not sure it's of the semantics here, but okay. And anyway, the highest risk factor for getting cancer, unfortunately, to say it in this room, is age. And it's the one that we really can't control. But in a genetic premature aging disorder, which is what I've focused on, cancer comes earlier than in the general population. Now, why hematology? I wanted one system. I wanted something, we use abbreviations a lot. You may have noticed this. If your doctor uses abbreviations, call them out on it. Say, thank you, could you tell me what you mean by that? Anyway, we learn about an H&P. That's the history and physical exam. And then the L is laboratory tests. And hematology, frankly, if I talk to the patient, if I examine them, and if I do some really simple lab tests, like a blood count and some chemistries or liver tests or kidney tests, I can make a diagnosis. And rarely do we have to go ahead and do the bone marrow test, which some of you may have heard of, or your children or grandchildren may have had. That's because that's where the money is. That's where the cells are made. It's in the bone marrow. So, inherited bone marrow failure syndromes. I'm a hematologist. I'm a specialist in cancer and genetic epidemiology. And I established a cohort, that's a group of families, where one or more individual, it might be their child, it might be they themselves, had an IBMFS. And the genetics of this is what I spent the last 20 years studying. And I'll show you, I have a slide, I think, on what it means for it to be a recessive or a dominant. And then what we did was we identified and quantified increased risks of cancer in these patients. So if the median age, and there's a difference, semantic here, between median and average. An average, all right, raise your hand if you're a mathematician. Okay, anyway, an average is to take all the numbers and sum them up and divide by the number of them. That's the average. The mean is the middle of that average range. The median is halfway through that, but it could be very skewed. It could be skewed towards younger or older. So in general, the median age of cancer is 70s and 80s, and in some of my populations, it was as early as 20. And we rarely had somebody over 30. So the genetic terminology, there are X chromosomes and Y chromosomes, and there's everything else, which are called autosomes. I didn't give the names to these things. It's hard enough to remember them. So an autosomal recessive means that each parent carried a mutated gene and they passed both of them on to the child. The X-linked recessive would be only in a male because males only have one X, the other chromosome is a Y. And in the male with the X-linked disease like hemophilia, that X has a mutation. The mother of a hemophiliac has two X's, one of which has the mutation and the other may be normal. So they may be normal. Homozygous means two copies of the same allele. It doesn't need to be the same mutation. So just each of the genes that would code for that protein has to be mutated. So Tay-Sachs would only occur if both parents pass a Tay-Sachs gene onto the child. Biallelic is just a funny word, a harder way to say, two variant alleles of the same gene, one from each parent. And a heterozygote, or wild type, and the wild type really comes from animal genetics. I don't think the parents who don't have the gene are wild. Well, they may be. But in any event, the heterozygote, or the carrier, has one mutated gene and one normal gene. So the types of disorders that I studied, in some cases, are named because of the doctor who described it, but there's no apostrophe S. They didn't have the... So Fanconi anemia, when I first started training, was Fanconi's apostrophe S, and I've had to throw out those slides because Dr. Fanconi did not have the disease. He may have described it, and he didn't even put the name on it, somebody else did. Diamond and Blackfan. Diamond was a pediatrician in the late 30s at across the river. Is it across the river? Boston Children's, yes. And Blackfan was the head of pediatrics. And Diamond was, and I don't know if anyone in this room besides me knows him, but he was not very humble. But nevertheless, his name was put on it because he described it, and he described it at a pediatric meeting that he had to be introduced by a more senior individual because he didn't belong to that pediatric society. So that's how Blackfan got his name on it, because he introduced him. Schwachman and Diamond is a different syndrome, and Harry Schwachman was a gastroenterologist at Boston Children's, and he started to notice that a lot of his patients who looked like cystic fibrosis but didn't have CF were having a problem with their white cells. So Harry Schwachman sent his fellow, Conti Ka, to see Lou Diamond's fellow in hematology, and who was, oh God, I'm blocking. But in any event, they didn't get their names on it. The senior doctors got their names on the syndrome. So I encourage all my trainees to make sure that they are the first author on whatever paper is relevant. Dyskeratosis congenita is another of these. It's the telomere disease, and it was first described at a dermatology meeting by Zinzer, Cole, and Engman, who were dermatologists, because the dyskeratosis means there's a problem with the skin. Keratin is the skin. Congenita means congenital. Well, from our perspective, they also have a problem with their bone marrow. So in many cases, we've abbreviated these words because it's really hard to give a talk saying all these words all the time. These are chromosomes, and you can see that some of them are intact, and I don't think my pointer works. But in any event, there are little arrows showing where the chromosome has been disrupted, and it's the cells, any cells, are cultured with a DNA cross-link agent called MMC, mitomycin C, or another one that causes breaks called diepoxybutane. You wonder why we abbreviate things? But these are the diagnostic tests. The recessive inheritance, I've written this for Fanconi anemia, which is a classical type of recessive. The red mark is the mutated part. So in each of these pedigrees, one of the grandparents is red, which means they have a mutated Fanconi gene, and their children can either get a normal gene or a mutated gene, and then the offspring, if they get two mutated genes, are affected because they don't have a normal gene. And then their children will have one affected gene and one normal gene for that same one, unless they happen to marry another Fanconi. And I mentioned Camp Sunshine, which is where families with children with diseases go, and we always worry about them becoming too friendly with somebody who has their same syndrome. This year, the camp, by the way, is being very careful, and they're making a little pod of each family. And so it doesn't matter what disease, they're not going to be interacting that closely. I have a statistician named Phil Rosenberg who I worked with for 20 years, and it was great fun. And one of the things he did was calculate the chance of marrying a carrier of somebody who has the same gene as they do. So if it's Fanc A or Fanc B, there's 23 of them, the chance is low. And we do have some Fanconi adults who've had children whose children are carriers but are not affected. So a dominant gene means that all it takes is one of them, as you see with the red, to be mutated, and that means that the offspring will have gotten the gene, and they may be affected. The X-linked, the affecteds are only male because they have only one X, and the mothers or female offspring are just carriers. Now, take a careful look at this picture. It's from 1974. It's a place in Italy called Bellagio, which is a Rockefeller-owned place that people can go for small meetings. And look really carefully, because there are only two women in that picture. Dr. Diamond's secretary and me. And I was there. I must say I owe to Dr. Diamond the fact that I do epidemiology, because I meant to bring it. You know what, you've all heard of spreadsheets. Well, you know what they are on the computer. Spreadsheets were big pieces of paper, and they were long columns, and you filled everything in, and I would go to those meetings having tabulated everything on his former patients. So I got to go to Bellagio, and then he had a meeting near San Francisco. So I mentioned telomeres. They're the repeats at the end of the chromosome, and when they're not there, they don't work well. And if you go on to their website, the company's called Repeat Diagnostics. The yellow will actually flash on and off, but I didn't know how to do that for this. Okay, so as you can see, I do a lot of teaching. Most of it, or a lot of it, at Camp Sunshine. the kids get to go to camp. This is for patients and siblings, and the parents and older patients get to hear lectures. And we also get to have meals together at tables of 8, just like here. Well, think of sitting there eating your breakfast and somebody shoves a blood count under your nose. That's inconvenient. So that's Camp Sunshine. It's really lovely, but not this year. And these are the doors. I fit in the middle door. And there's a family showing what the different doors are for. And then, because it's for children, there's Sonny the Bear, Chip Chip the Chipmunk, Moxie the Moose, and Loopy the Bear. And the kids have a great time. And how many of you seen Sully and Mike? Not a lot. Well, Sully's left arm gets broken a lot because the kids like to swing on it. But sometimes they're small enough, as you see. So where have I been? I've been to a lot of countries, and the ones in italics are the ones I went to for vacation to get nice pictures of birds and animals, and the non-italic are the other countries that I've been to for work, and some of them I've been to both. And one of the nice things I like about all this travel is that I usually have colleagues in these countries, and I get to see things from a different perspective than just being a visitor. It really makes a huge difference. And where am I going and when? Stay tuned. Thank you. You're speechless, I know, but you lost your microphone? I think you gave it to Mike. Gave the mic to Mike.
Anybody have questions?
First of all, thank you. You briefly talked about DNA, RNA. I'm curious what you can tell me about the mRNA technology.
Oy. More of a, M stands for messenger. So the RNA is called a messenger. There's two types of RNA. There's messenger RNA, which takes the message from the DNA and helps to form a protein. And then there's also other kinds of RNA that are involved in making the RNA that is then used for that purpose. And on a need-to-know basis,
I don't know if this relates to your profession or not. My daughter, when she was 27, had breast cancer. 5 of the nurses that she worked at the Children's Hospital, all got breast cancer, all 5 of them. Thank you, God, they're all fine now. But I was wondering if that has any relations to any of the things that you were discussing.
So, first of all, in their case, probably not. One of the many genes involved in the Fanconi anemia DNA repair pathway is BRCA2, and the other one's BRCA1. But they are minor genes as far as the Fanconi pathway, because you have to have a mutation in both. So I will tell you an anecdote, which is the group I worked with at the Cancer Institute is called the Clinical Genetics Branch. And at one of our meetings, one of the people said, you know, I'm following up on these breast cancer families that are enrolled in our study. And one of them, when I mentioned the description of the birth defects in Fanconi anemia, said, oh, that's my grandchild. And so breast cancer may occur in somebody who has a mutation in BRCA1 or 2, and then the other gene may not be mutated, but may be subject to damage or something. Whereas to have Fanconi anemia, you have to have a mutation in both of those genes. But it is one of the genes that we have studied. It's also, the Fanconi genes were started out as A, B, C, D1, D2, because they misclassified, etc. And FANC D1 is BRCA2. That much I can remember. But thank you. But the fact there were so many people, unless they were all Jewish, there are mutations within BRCA1 and 2 that are Ashkenazi Jewish mutations, but there are also people who do not have that background and have different mutations within the same genes. So it's, sorry, it's not a contagious disease, as far as we know. Oh, yes.
Okay, you mentioned, I guess it was telomeres or
Telomeres.
telomeres, okay. And there's been something in the news about ways of lengthening those, and increasing.
Well, okay. Telomeres are shortened repeats at the end of the DNA within the chromosome. I'm not sure that there's something bona fide that would help to lengthen them. There's going to be a lot of literature on that online, but it's not something that I have any insight into. And I'm not assuming that my telomeres are going to grow. I've given up growing a long time ago. Yes.
I have a question. When you stated in the beginning that cancer usually affects the aged person, could you just talk about the studies that were done for that? Because I've never heard that before.
I'm a little surprised. It's, I suggest, I mean, Fanconi anemia, which is one of the disorders we study, we consider a premature aging syndrome.
Okay.
There are several of those, which are showing at an earlier age things associated with the normal aging process. The biggest risk factor for cancer, if you look at any of the NCI data, is age. And that's why the risk of cancer is higher as you get older. And what we have described in Fanconi anemia, also in dyskeratosis congenita, Diamond-Blackfan anemia, even Shwachman-Diamond syndrome, is some of the kinds of cancers that occur in these patients at younger ages. And so, for example, Fanconi anemia is a very highly cancer-prone disorder, but the cancers are very specific. They're usually head and neck and throat cancer and sometimes gynecologic cancer, which is totally different from the kinds of cancers associated with getting older. But that's part of what we studied and identified the types of cancers and the relative ages and the relative risk with age. But overall, at any age, it's probably a 300-fold increased risk compared to people who don't have two mutations in Fanconi.
Excuse me, I'm trying to understand that. Does this mean if there's been something in the family before as part of the family?
No, not necessarily.
Okay.
Because cancer might be due to environmental factors, smoking, radiation, all sorts of things. So it doesn't, and those may not be familial, they may be environmental.
Okay, thank you.
Yeah. Faye, you get your exercise.
Okay, so I don't completely understand about RNA. I mean, I understand what DNA is. What is the significance of RNA, and is it part of DNA?
No, DNA has to be converted into RNA to make a protein. DNA per se carries the genetic information, but it cannot turn into a protein.
Why should it turn into a protein?
It can't.
But why would you want it to?
Well, but that's what RNA does.
But the point is, what relevance does it have to us?
The only relevance is that you're all familiar with the fact that you're getting vaccinated here with an RNA vaccine, which is somewhere between DNA and protein. It's just a biologic thing. You don't need to, on a need-to-know basis, you can't control it. Yes.
Hi there. During the time you've been studying these syndromes, have you witnessed any breakthroughs in treatments or in cures?
Well, as I say, I'm an epidemiologist, so I'm a bean counter. But certainly in the time that I've been involved, for example, bone marrow transplant can cure anybody at any age who has what's called aplastic anemia, inability of the bone marrow to make blood, or leukemia if they have an appropriate donor. So that really has developed. When I was a trainee at Boston Children's, I won't do the math, a few years ago, they were first developing bone marrow transplant. And at that time, a bone marrow transplant was done in a special unit in the hospital called a clinical research center, and it was a big deal. And now it's done routinely in many places very frequently. Yeah, so that would be probably the major thing. The other thing is that we can now, because of our studies, we know what the patients should look out for. So, for example, the most common solid tumor in Fanconi anemia is head and neck and throat cancer. And we've got patients now enrolled on screening protocols, which didn't used to happen. They'd show up with something that was already advanced. So, I think that Fanconi anemia is the example of a DNA repair disorder. Dyskeratosis congenita ultimately is a problem with broken DNA as well. And what we did is studied the 4 syndromes that I've mentioned. Diamond-Blackfan anemia, for example, have different kinds of solid tumors than Fanconi or dyskeratosis. And Schwachman-Diamond is controversial, but it seems that if they have solid tumors, it's a very reduced risk. But all of these disorders are at risk of leukemia, which is why we do blood counts and try to convince families to let us do routine bone marrow tests as well.
Blanche, that was a fantastic talk.
You like my moose and meese?
Absolutely. So,
He speaks my language.
so the question that you had about cancer and aging, I'd like you to comment on, we all know that we're all exposed to environmental factors. And so we're probably getting cancers throughout our life, but they never manifest because our immune system clears it away. And as we get old, our immune systems age too and become less effective. Would you like to comment on that?
Well, so what Michael is trying to suggest is that the higher risk for cancer with aging is due to a change in the immune system. Maybe. Thank you. Implication. Because in fact, the types of cancers that I'm indicating are occurring statistically at a much younger age in my patients. So tongue cancer, how many of you, of course you all go to a dentist, and of course they move your tongue around and they look in there and they ask about a sore throat. I'm not asking for a show of hands of those who don't undertake this process, but they're doing it because they're trained to do that. But they're not expecting a 20-year-old who walks in to have a tongue cancer. Whereas amongst my population, unfortunately, if a patient who's 20 and comes in and says, I got this sore in my mouth, here you think of, they bit their cheek or it's a canker sore. If they have Fanconi anemia, I think about cancer. So, unfortunately, back there.
Let's see if she still has her question. Did you start your question?
I should have given out numbers.
I had a question that is non-scientific. It deals with your career. You, in 1974 or whenever it was, you said you were the only woman in the room aside from a secretary. How has that changed and how has it changed?
That's a very interesting question. Let me rephrase it. The question had to do with the fact that there weren't a lot of women at the meeting in Bellagio. Not only that, but the men who brought their wives for a tour of Europe, the wife had to stay in the village in an inn and wasn't even allowed to stay at the Rockefeller property. But medical schools now are slightly in excess of women. So what I didn't show, and I should have put that in here for that, to head that question off. I have a picture of my medical school class. How many women do you think were in it? Well, there were 5. Yeah. And because they did a class picture, I used to have that on the wall in my office, and when young people came in, I said, you know, pick out the women in here. And they could never find all of them, even though 3 of us were sitting next to each other. So, but that's changed. It's a slight excess of women. When I worked at Mount Sinai Hospital, the guy in pediatrics who was choosing the next intern class called me about one of the candidates who had worked for me as a lab tech. And I said, well, that's interesting. Why are you asking about him? He said, well, he's a he. Because the majority of the applicants, even then, were women. And pediatrics is still predominantly women. And those of you who've had children might have preferred a woman on the perception that they might be more sensitive. So I'm going to tell you an anecdote that I could only tell at Newbridge. When I grew up during the Second World War in that other country, the head of pediatrics at the Hospital for Sick Children in Toronto, known as Hospital for Sick Kids, had all the Jewish mothers giving their kids bacon because we needed a source of protein, and that was all that was available. And for those of you who have the background with enough to know that it's okay to do whatever you have to do for health. You can ignore those 600 rules. And my mother chose to ignore them, and her father, who was Orthodox, would take a long walk because he didn't want to smell the bacon. So somebody back in the back has their hand up.
I'm just getting back to Faye's question, because we all learned about DNA in high school biology a long time ago, and we think of it as what defines characteristics.
Most of us didn't even learn about DNA in high school.
Mendel's
College.
theory, I thought that was DNA.
Yes.
But in any case, we think of it as defining characteristics.
Right.
And what you're talking about is proteins. And
Well,
we don't-
because the
I don't
DNA
know
is where
what
the mutation is.
Right. But what's the importance of proteins? Because you need to have
protein is the
RNA to
active
have the
component
protein.
that does the job. So if the DNA is mutated, it can't make a normal protein. If the protein is mutated because the gene was mutated, then you have the disease.
Oh.
So protein does the job, and DNA tells it what to do.
Okay.
What's that? The supervisor, thank you. And this is from a physicist. Very good. Thank you. There's somebody in the way back there. Bob knows where to find me. So I'm just curious, because we're all of similar age, how many recall learning about DNA in high school? Well, you don't count. Well, I didn't. I didn't learn about DNA till halfway through college. It wasn't called DNA?
I don't know that they knew about DNA, but it was, they were just genes.
There were genes, yeah, but they didn't know what the gene was made of. Yeah, yeah. That was where Watson and Crick got together. And that was when I was in college and literally took a course. I dropped Watson's course because it was bacterial viruses, but I took Crick's course a couple of years later because it was on DNA. Yeah.
I hope you find this amusing. I was a sick baby. I had bronchitis, I guess, quite often until I was even just a young child. And my mother came from an Orthodox home. She was Orthodox, kept a kosher home. And the doctor, and I don't remember who it was, that she was using as a pediatrician at the time, told her that to feed me bacon.
Which city was that in?
At that time, I was living in, I think it was Roxbury, actually.
Okay.
I can't tell you who the doctor was.
Well, let me tell you that Lou Diamond of Diamond Black Fan Anemia was Jewish. Schwachman was Jewish. So, and they weren't GI docs anyway.
Yeah, but I
Yeah.
thought you might.
So that's where my doctor was Dr. Alan Brown. And it was clear that he only told the Jewish families to take bacon. Right.
Are you familiar with amyloid?
With what?
Amyloid or amyloidosis?
Not enough to answer any specific question. It's concern about whether it's involved in Parkinson or in aging, but that's all I know. My patients don't live to get old enough for that to be their major problem. Did you have your hand up? No. Okay.
So I could go back to Bob
Oh,
now?
Bob.
Okay.
Bob has been waiting patiently.
Finally, same question.
I'm steering clear. I don't want to be tripped.
So lest you think that it's only her patients with these hematologic conditions that get cancers, it occurs in almost all specialties.
Put your mic closer.
What?
Yeah.
Oh, okay. So I'm a kidney specialist, as most of you know, and back in 1978, I found with my fellow, and had my fellow be the first author, by the way, the first inherited kidney cancer in adults. And Wilms tumor was known in children. So take kidney cancer. You asked about age. So to get a cancer, your normal cells have to be changed. And they have to be changed so that they can reproduce. As we mentioned, the body is mainly proteins. Proteins work on muscles, our organs, everything pretty much that we do, all the enzymes, they're all proteins. But to reproduce, you got to work at the DNA level because they're going to make new cells. And you need to damage them in a way that they will reproduce abnormally. So as you age, the environmental factors you're exposed to produce all kinds of damage. We try and repair, but if it doesn't work, you can end up getting a cancer. It requires at least more than one hit, probably two or more at least. So say in our kidney case, we found a family that has an alteration, as she showed the chromosomes, where one chromosome had separated and joined another, and vice versa, so that they had two abnormalities. They had all their genes, everything was normal. If you looked at this family, they looked normal in every respect. So kidney cancer, the average age is around 61. That would, if you're going to get it, that would be the average where you've had enough damage to kidney cells that maybe you'll get a kidney cancer. In this family, more likely in their 30s or 40. And the reason is they start with a defect on one side, a tendency for the cells not to reproduce properly. We know that, we didn't know that at the time, we just knew they had this marker, later finding that the marker tended to delete a few genes. So they lost some, and then if they get damage on the other chromosome, they could end up with a cancer. And like your patients, we followed them with ultrasounds. At the beginning, I had to get the companies to pay for ultrasounds. They said, I'm not going to pay for an ultrasound on a healthy person. I said, well, you'll certainly pay after they get a cancer, and it wasn't detected. So they'd knuckle under and do it, and we end up saving saving a lot of lives the same way you've done with
Well, basically,
the
the po-
hematologic
The
disorder.
point of our cohort was, we started with people who had blood diseases and identified that they were at risk of various cancers and of what age and what type, and therefore could offer prophylactic screening, if you will. Yeah,
You did really good work.
thank you. Thank you. I'll pass that on to my colleague who's still at the NCI and tell her that people want her to keep going.
Blanche, thank you.
What's that?
Aren't you glad you took his question?
Yes,
Thank
Michael.
you so much. That was a wonderful talk.
Thank you.